So this is the review for your test 2. Just as a reminder, I wanted to let you know that it's 15% of your final grade, and you have a final exam, which is coming up on December 9th at 2 to 5 p.m. in the gym, which is 30% of the final grade. The test 2 is on November 14th, so that should be Monday evening from 6:30 to 8 p.m., and that's in Huggins 10. And anything that was mentioned in class may be tested. This review should help you focus your studying. However, anything that was discussed in class you may be responsible for. The exam, this particular exam is about 27 multiple choice questions and then short answer questions which make up 27 points, so approximately 50-50 multiple choice and short answer. What are exergonic and endergonic reactions? So we were looking at metabolism, and there's a few terms that you'll need to be familiar with for understanding metabolism. Firstly, what exergonic and endergonic reactions are. So exergonic reactions occur when you have the release of energy, and that happens when you have a breakdown of a molecule, and that is catabolism. Anabolism is when you have the generation of a new molecule, so you're building a molecule, and that requires energy. So that's an endergonic reaction, whereby it requires energy for it to proceed. And most of the time, these reactions would be coupled, so that the energy released from the breaking one bond or breaking down one molecule can be used to build another molecule. So they're coupled. What are photoautotrophs? These are photosynthetic organisms, and they can generate ATP from harnessing light energy, and then they can use that energy that they get to make carbohydrates. So they can take carbon source, which is often carbon dioxide, and they can generate carbohydrates with it, so they can make glucose. Often this results in the production of oxygen as well, depending on the microbe. Can all prokaryotes live in the presence of oxygen? Why or why not? So we talked about a few examples of prokaryotes and how they live with or without oxygen and the names that we would call them. So if there's a microbe that has to grow in oxygen, we would call it an obligate aerobe. And this microbe would be found at the top of a tube. A obligate anaerobe or a strict anaerobe will be found at the bottom of the tube, so furthest away from any oxygen as it can get. In contrast, a facultative anaerobe will be found throughout the tube and a lot at the top of the tube. The term facultative anaerobe means that it can grow with or without oxygen, and it actually grows better with oxygen, and that's why you find it at the top of the tube here. An aerotolerant anaerobe doesn't need oxygen and can grow with or without it, so you may find it anywhere throughout the tube. And then a microaerophile needs a small amount of oxygen, but not the same amount as what you'd find at the top, so you often find it growing in a layer close to the surface. And part of where you find these microbes growing is based on their enzyme content, so whether or not they have superoxide dismutase and catalase. So these are enzymes that can detoxify toxic forms of oxygen. And so in order to grow with oxygen around, they need to have at least some of these enzymes around. So the obligate aerobe has these enzymes, the facultative anaerobe has them, the aerotolerant has some, the strict anaerobe would often not have these enzymes, and then the microaerophile would have some of the enzymes, but not always all of them. what happens during oxidation reduction reactions? So we talked about several different oxidation reactions and the cofactors that are used in metabolism. Oxidation is loss of electrons, and reduction is gain of electrons. So this is an example of an oxidation reduction reaction. So one molecule will lose electrons and the other will gain it. So in this example, compound A is oxidized in this process and B gets reduced because it gains an electron. Why does fermentation occur? So we looked at fermentation a little bit, and what I want you to know from fermentation is that it is important for microbes in order to continue getting some energy when aerobic respiration can't proceed. So you would get, the cell would need to, in order to survive, it would need some ATP generated, and you get 2 ATPs, 2 net, through glycolysis. And so in order for glycolysis to continue, so this is an example of glycolysis, some of the steps, you need a recycling of the NAD. So you need to have NADH oxidized to NAD that can be used in glycolysis, so glycolysis can continue. So fermentation will give you, depending on the microbe, different types of products. One of them can be a lactate, and this is important so that you can have the recycling of these cofactors that are needed for the cell to have glycolysis to continue. Because that's where you get some ATP under anaerobic conditions. So when there's no oxygen around and there's no shuttling of pyruvate into the citric acid cycle and ultimately to put electrons and protons into the electron transport chain. So what is gained from glycolysis? So what are the products? Well, what are the starting points? You start with glucose. You also need a few other things for the pathway to proceed. You need some ADP, you need inorganic phosphate, and you need NAD. So that's the cofactor that you need. And because you can store energy in the bonds by adding hydrogens here. So in breaking down glucose, that's a 6-carbon molecule down into 3 carbon molecules, into 2 3-carbon molecules, pyruvate. In this process, you generate 2 pyruvate from 1 glucose. You get 2 ATP, and you get 2 NADH. And so you actually, it takes 2 ATP to get this process started, and then you get 4 out. But then you would have a net of 2 ATP overall. And you get 2 NADH because you have NAD being reduced to NADH here, as the glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. And this happens twice because you have the breakdown of glucose into 2 3-carbon molecules. So where is CO2 released? CO2 is released during the Krebs cycle, which you may hear it also called the citric acid cycle, or the TCA cycle, or the tricarboxylic acid cycle. And so in the citric acid cycle, you have pyruvate coming in as acetyl-CoA. So before it gets to the cycle, this is the cycle here, it is actually, pyruvate is decarboxylated, so there's a removal of carbon dioxide from it. And in this process, you get the reduction of another NAD to NADH. And so this would happen twice per glucose molecule. So you have the pyruvate being converted to acetyl-CoA, and in this process, it's then going to shuttle it to the citric acid cycle. So acetyl-CoA combines with oxaloacetate to give you citrate, and then This is isomerized to isocitric acid, and then this is decarboxylated, and in this process you get the reduction of NAD to NADH, which is where you will get a lot of energy when this is moved into the electron transport chain. Then you have the when it goes from isocitric acid to alpha-ketoglutarate, you have another decarboxylation step, and you have NADH generated again. Then it gives you succinyl-CoA, and when you're going from succinyl-CoA to succinate, this gives you an ATP. So it could be an ATP or a GTP, depending on the cell. Overall, this is a way to get some energy from the citric acid cycle. Then you have the reduction of FAD to FADH2, which is another cofactor that's an electron acceptor, and you generate fumarate. And fumarate is converted to malate, and malate is converted to oxaloacetate. So that's the starting of the cycle again. And you have, in that process, you get another NADH generated. So overall, with one turn of the cycle, you get 3 NADH. That's not including this NADH up here from generating acetyl-CoA. So you get 1, 2, 3 NADH, you get 1 FADH2, and you get 1 ATP. The citric acid cycle happens in the inner mitochondrial matrix of eukaryotic cells, and it occurs in the cytoplasm of prokaryotic cells. Where does the Krebs cycle occur? Oh, I just answered that question. It occurs in the inner mitochondrial matrix of eukaryotic cells, and it occurs in the cytoplasm of prokaryotic cells. What happens to NADH in the electron transport chain? Where does NADH come from? And where is the electron transport chain in eukaryotes versus prokaryotes? So NADH in the electron transport chain gets oxidized to NAD. So that's what happens to NADH. And the NADH comes from, there's 2 from glycolysis, and the other ones are from the citric acid cycle. And this occurs, the electron transport chain occurs in the inner mitochondrial membrane of eukaryotic cells, and in prokaryotes it occurs in the cell membrane. So in the generation of ATP, you have this process where NADH gets oxidized to NAD, and then there's this pumping of protons across this membrane. And this is complex 1 is where the first, or where protons are pumped from getting a movement of electrons from NADH. And so FADH2, it joins or enters in complex 2 here. And so you don't have as many ATP generated from FADH2 because it doesn't move as many protons across the membrane. You have protons being moved across the membrane in complex 1, 3, and 4, but not in complex 2. So you have the movement of these electrons through complex 1 to coenzyme Q, and then to complex 3, or from complex 2 to coenzyme Q to complex 3. Then in complex 3, you have the movement of more protons outside of this cell membrane, and then the movement of electrons to the cytochrome C. And then you have the movement of electrons through complex 4, and you have the reduction of oxygen to water. And in here you have some more protons pumping. So overall you end up getting a whole bunch of positive charges over here because you have all these hydrogen ions with this positive charge. And so it builds up to be all this positive charge over here, and then they go down the concentration gradient through the ATP synthase, and they will move this ATP synthase like a motor, which generates ATP. So you have the generation of ATP, which is dependent on this proton gradient that's formed, and this is true for both prokaryotes and eukaryotes. What is the final electron acceptor in aerobic versus anaerobic respiration? So we talked about how in aerobic respiration the final electron acceptor is oxygen, and so that's here. Oxygen is the final electron acceptor. In anaerobic respiration, you can have other final electron acceptors. You won't get as much energy from it as you would if it was aerobic respiration, but there are microbes that can use other final electron acceptors such as nitrate, sulfate, carbon dioxide, iron, just as a few examples. What affects the growth of bacteria? What are the names of microbes that like to grow at different temperatures? So the effects of what affects the growth of bacteria? We talked about a few different things that can affect the growth of bacteria, and specifically the first one would be temperature. And so microbes grow at different temperatures optimally, and depending on the temperature, we give them a different name. So you don't need to memorize the exact window, but have an idea of the middle of the window. So the psychrophiles, the middle of the window is close to 4 degrees, so in the fridge, maybe a little bit higher, but it can grow in the fridge. Mesophiles grow best at around 37 degrees, and so they would grow in our bodies quite well. So most human pathogens would be mesophiles. If they grow best around 60 or 65 degrees Celsius, they would be considered thermophiles, and if they grow at temperatures even higher, 90 or 100 degrees, they will be hyperthermophiles. What happens during bacterial growth? So during bacterial growth, if we look at the number of bacterial cells over time, they initially have this lag phase. And so in the lag phase, it's when they're just starting to realize that they have new nutrients, and it takes them some time to kind of clue into that and start dividing. And then they grow and divide exponentially while the nutrients are optimal. And so they will divide, depending on the microbe, every 10 or 20 minutes. Then they reach kind of a plateau. So this is the stationary phase, where the number of bacteria that are dividing are equal to the number of the bacteria that are dying. So you don't see any growth and you don't see any death yet. And then there comes a point when you see a decrease in the number of cells, because the number of cells that are dying are greater than the number of cells that are dividing. So you should know these terms and what's happening with the bacteria under each of these terms. How long will it take to get to a number of bacteria if it doubles every so many minutes, if there are so many bacteria to start? So this is an example we did in class. So let's say this particular bacteria divides every 20 minutes, and let's say we start off with one bacterial cell. So if I ask you how long will it take to get to 16 bacteria, you should be able to tell me it would take 80 minutes. So if we have one bacterial cell and it divides every 20 minutes, the one in 20 minutes later will become 2, and then another 20 minutes, those 2 will become 4, and then in another 20 minutes, those 4 will become 8, and then in another 20 minutes, those 8 will become 16. So it'll take 80 minutes to get to 16 bacteria. Calculate how many bacteria are in a sample. This is something I think you should have done in the lab, but we also talked about in class with respect to calculating how many bacteria you're counting from a plate, so how many CFUs are on the plate, colony forming units on the plate. And how I do it is, may not be how you do it, but as long as you get to the same answer, it shouldn't matter, and as long as you know your units. So if I had a plate of bacteria, let's say I counted 40 colonies on that plate. So that's in a good range. It's more than 20 or 30, and it's not too high, so it's a good count. So I'm going to say that there's 40 CFUs on my plate. And then I'm going to times it by the dilution factor of the tube, but I need to take the negative out because I have this on the top of my equation here. So I would times this by the dilution factor of the tube that I took this and put some of it onto this plate. So if I took, let's say, 0.1 mils, and spread it out onto this plate, and I took it from a tube that was a 1 in 100 dilution, which is the same as 10 to the negative 2. So what I would do here, I would, if I took from a dilution tube that was 10 to the negative 2, make that negative bigger, and I took 0.1 mils of it and I spread it out onto this plate, I would then come here and I would say that I times it by 10 to the 2, and so I take out the negatives because I have this on the top here. And then I would divide it by the amount of mils that I put on the plate. And so I put 0.1 mils on this plate to spread it out. So I would put 0.1 mils here, and that's where you get your units from. So I would then do the calculation whereby I would go, this gives me 40, and then 2 zeros from this, because this is 100, and then I add another 0 from this. So if you have a calculator, it should give you 40,000 CFUs per mil. So as long as you, there's many ways of doing this, but you should get to the same result in the end. And that means that in the original tube there were 40,000 CFUs per mil. Okay. What effect does oxygen have on bacterial growth? If you look at the growth of bacteria in a chart and the effect of oxygen, you should be able to draw a growth curve, so of bacteria that are grown with or without oxygen, so or be able to recognize what it looks like. So this is the number of bacterial cells over time. So time is on the x and number of cells on the y. And in the first graph here, it's showing us how the bacteria that are aerobic, so they want to grow in the presence of oxygen, and we are growing them in this example in the presence of oxygen, how fast they grow. And they grow quite fast. That's a steep curve. Then it's the second picture, it's showing us how there is an anaerobe growing in the absence of oxygen. So it's anaerobic, it wants to grow without oxygen, and if we grow it in the absence of oxygen, this is the growth curve that you would see. So the growth curve would never be as steep as the aerobic bacteria growing with oxygen, because you don't get as much energy in metabolism through anaerobic respiration as what you would for aerobic respiration. So that's why this one is steep and this one is not as steep. Then if we considered the facultative anaerobe, so that's the one that can grow with or without oxygen. It actually grows better with oxygen because of the amount of energy you get when oxygen is around. So there's a lot of it at the top of that tube. So we have a facultative anaerobe growing in a cup exposed to increasing amounts of oxygen. So you have oxygen around. That's growing faster, so the curve is steeper. Then the same facultative anaerobe, or same type of bacteria, growing with a reduced amount of oxygen. So this is one that students often get confused about because the name anaerobe, they often think it wants to grow without oxygen and it would grow only without oxygen, but that's not true. The term facultative anaerobe means that it will grow with or without oxygen, and it actually grows best with oxygen. What is a biofilm? So we looked at biofilms. They can exist on rocks, they can exist in your toilet or on your shower, and they're this clumping of bacteria, and they kind of develop this community. And in this community, the ones that are deeper into the biofilm will be growing anaerobically, so they'd have, or at least they'd have less oxygen than the ones closer to the surface. And they'd also be growing slower because they have less access to nutrients. They will also kind of have the photosynthetic bacteria in the biofilm at the top layer, and the non-photosynthetic ones deeper in the layer if there are photosynthetic ones there. So they kind of move or generate their communities so they're beneficial for the communities. They can attach to different surfaces, and they can attach to any conditioned surface. So then we looked at biogeochemical signaling. What is the relevance of biogeochemical signaling? Well, those biogeochemical, I think it's supposed to say cycling. The biogeochemical cycles are important because they are needed for the movement of elements that are essential for the building blocks of a cell. So we talked about how phosphorus is important for things like the lipid bilayer. Phosphorus is needed for DNA. Carbons are needed for lipids, carbohydrates, ATP, carbons, and everything. So all these elements that we looked at are essential for the building blocks of a cell. What are the microbial processes that occur in the nitrogen, carbon, phosphorus, and sulfur cycles? So you'll need to know the parts of these cycles that, with respect to that bacteria are involved in, and if it's getting oxidized or reduced. So they, for example here, ammonia is getting getting oxidized to nitrite and then to nitrate. So you'll need to know these cycles, particularly the nitrogen one, but you will be responsible for all of them. You may look at the other ones in the slides though, with respect to that were given in class. What is lake eutrophication? So this is a question that I like. It's because I think it's kind of neat to see what can happen to a lake. I don't think it's really neat. It's actually quite disturbing. But when there's pollution in a lake, so if there's pollution that's run off into a lake, you get this excess of inorganic nutrients that wouldn't normally be there. And so those nutrients that are there, particularly nitrogen and phosphorus, can cause problems if they are in excess in the lake where they wouldn't normally be. You get an algal and cyanobacteria growth. So you get an algal bloom and a cyanobacteria growing. And these two microbes will generate a lot of oxygen, which you might think oxygen is good, but over the long term, that means that if the lake gets pumped full of lots of oxygen, because all these microbes are growing excessively and making so much oxygen, it allows for an overgrowth of bacteria that use oxygen. So aerobic bacteria will grow excessively, and they will ultimately deplete the oxygen that are in the lake. So this takes time for this to occur, but it is very detrimental to the lake because the animals and fish and other species of organisms that live in that lake are going to die because they're not going to have any oxygen anymore because the aerobic bacteria are going to use it all up. And then when you have anaerobic conditions, so the lake is depleted of oxygen, you have anaerobic bacteria growing, and they can use in anaerobic respiration other final electron acceptors other than oxygen, and so they can use, for example, sulfate, and they can ultimately generate excess hydrogen sulfide, which is very stinky and will make this lake not smell good. So this is a process that happens after there is pollution into a lake, and it can destroy that lake. So you should know the steps and why one step kind of causes or feeds into the next one. What effects do living things have on each other? We talked about how there are these microbial interactions and how they have relationships that can be intermittent or cyclical or permanent. And so they may be given the name or term mutualism if there are two organisms that are mutually benefiting one another. It would be called cooperation if one of the organism is positively benefiting the other, but it's not obligatory for that organism to survive. So in mutualism, they are providing something that is obligatory. In commensalism, there's a positive effect on one organism, and no positive or negative on the other. In predation, you have one organism that's the predator and one that's the prey, and it could be a bigger or smaller, depending on the example. One example would be that protozoa, which are bigger, are predators of bacteria. They're prey, and they can eat them and destroy them. It would be parasitism if a parasite comes in and infects a host and causes problems for that host. It's called amensalism if there is something secreted from one organism that has a negative effect on another organism. And competition. They may be competing for certain resources, and this may result in one of the organisms growing and the other one not being able to grow, or they may both exist at lower levels than they would otherwise. What is the sequence of events that need to occur for microbial disease to develop? So this is looking at considering that you need to be contaminated with an organism before you get an infection. And then in an infection, that's when the pathogen is growing in an individual, and then it would be considered a disease after that. What is a zoonotic disease? So we looked at some examples of where you would get an infection from an animal transferred to a human. And so this is a large percent of infections are caused by zoonotic diseases. So this is transferring a microbe from an animal to a human. And one example is the hantavirus, which is passed through mouse deer droppings to humans. So sources of pathogens can be from other living things, and it's a zoonosis or a zoonotic disease if it's from an animal. When is it possible to eradicate a microbial disease? So you can eradicate a microbial disease when and if that microbe exists in one reservoir. So if the microbe only exists in humans, for example, it's possible to eradicate it through vaccination and isolating any individuals who do get sick from other healthy individuals. So the example we talked about in class was smallpox. And smallpox is thought to be eradicated because of the fact that it has only a human reservoir, and we were able to vaccinate the majority of populations until smallpox died out. What are the modes of transmitting microbes? So you can transmit microbes from one person to another, so through direct contact. So it can be direct contact, it can be through airborne droplets, or vertical contact is considered from a mother to a fetus. You can have transmission of an infectious disease through a vector. So a vector could be, for example, a mosquito. And a mosquito, it would be a biological vector if that mosquito has if the microbe has part of its life cycle in the mosquito. It would be considered a mechanical vector if it's just moving a microbe from one location to another. Indirect vehicles would be inanimate ways of contracting a microbe or an infection. And so they can be through contact with fomites, so things that have microbes on them. And this is a picture of a door handle. It could be a towel, so towels at the gym, things like that. It also could get indirect transfer from food or water, or could be airborne. And so you can get microbes that exist in the air. This car is, for example, kind of making lots of dust, and so kicking up any of the microbes that exist in the dirt and can cause someone a problem if they breathe those dust particles in, especially if there's fungal spores in there, and someone who breathes it in who is immunocompromised. How do normal flora help prevent infection? They are helpful in preventing infection because they make it so there's no room for the microbe or a pathogen to kind of gain or get nutrients from the host. So one example is, I don't know if I told you guys this, but there are chickens, for example, that when they're born, so when baby chicks are born, they actually spray them now, not all the time, but with this thing called Preempt. And the idea is to prevent these chicks from getting Campylobacter or Salmonella as part of their normal flora by spraying them with these, normal flora that you kind of want them to have, so that they get colonized when they're born with a normal flora that wouldn't make us sick. So if they don't have any room for Campylobacter or Salmonella to grow on them, they won't. So maybe it would be helpful for us in the long run. There are terms that we use with respect to a pathogen. A pathogen may be always a pathogen, so it causes a disease every time it comes in contact with the host, or may be opportunistic, whereby it gets causes an infection when it gets the opportunity to do so. So if it's part of the normal flora, it may cause an infection if it gets into a different tissue. So for example, in the microbes that exist in your gut, they're fine in your gut, but if you were to vomit and aspirate that into your lungs, they could cause a pretty bad lung infection. So if they get the opportunity to go to a different location, they can make you sick at that different location. What is an endotoxin versus exotoxin, and what are their mechanisms of action? So we talked about endotoxin before. Endotoxin is in the cell wall of gram-negative bacteria. Exotoxins are released from bacteria, and they can be released from gram-positive or gram-negative, depending on the microbe. And they have different mechanisms of action, so in causing effects on the host cells. If we look at exotoxins, not all, but a lot of exotoxins are these AB exotoxins. And so they have effects on the host cell. And the A is for the activity and the B is for binding. So the B binds to the host cell to get in. And so the example that I told you guys about is the cholera toxin. And so it's not here, but it's in a chart, and it shows you that it causes, once it gets into the cell, the A part in cholera toxin actually causes the secretion of water. So water comes out of this cell, if it was an intestinal cell, and causes a lot of diarrhea. So not all exotoxins cause cell death, but some do. This is another example of an AB toxin mechanism of action. One of the ones that I wanted to highlight was the cholera toxin and how it causes diarrhea by causing the water to be released from that cell. Endotoxins are the toxic part of lipopolysaccharide, so lipid A. And this is in the cell wall of gram-negative bacteria. They can have effects on host cells by interacting with innate immune receptors, so these pattern recognition receptors that are on the surface of host cells, such as these phagocytic cells called macrophages. And if there's endotoxin comes in contact with its receptor, which is TLR, one of the receptors for LPS is TLR4, and this can signal into the cell to tell the cell to change its gene transcription and to activate the production of cytokines. So cytokines can have many effects. They can recruit other cells to the area. They can make the vasculature increase in diameter and become leaky. So blood and complement proteins can come into the site. They can also cause a drop in blood pressure and change your temperature, and induce fever. And actually, fever is actually helpful in killing some microbes. So sometimes a little bit of a fever is a good thing, but overall, a high fever is not healthy for an individual, not for too long anyway. What cells are involved in the innate immune response versus the adaptive immune response? So we looked at a lot of cells, and you will need to know all of them. This is a highlight of a few of the major ones. So in the innate immune response, some of the major ones, the major players that we looked at were phagocytic cells such as neutrophils and macrophages. And so they're really good at phagocytosing, which means eating and destroying microbes that they find. We also looked at dendritic cells, which are part of the innate immune response, and they're actually really important in linking the innate to the adaptive, because they are a professional antigen-presenting cell. With respect to antigen-presenting cells, dendritic cells can do it, macrophages can, and so can B cells. So this dendritic cell, if it finds bacteria, say, or a pathogen that came in a cut in the skin, it will go to the local draining lymph node and present what it's found in the context of MHC to the T cells in the local draining lymph node. And so the T cells and B cells that you find a lot of them in the lymph nodes are lymphocytes. So lymphocytes are from a lymphoid progenitor cell, and the major kinds are T cells and B cells. And we talked about T helper cells, which help by producing cytokines. And we talked about cytotoxic T cells, which role are to kill infected cells. So they kill host cells that are infected with a microbe. The B cell's main role is to produce and secrete a lot of antibody, and this is needed to neutralize or phagocytose and destroy or activate complement, ultimately to get rid of microbes. Then we looked at some of the innate immune receptors. So what do the toll-like receptors do, and what's the difference between TLR4, 2, and 3? There are many more toll-like receptors than that, but I want you to note these three. The toll-like receptors are innate immune receptors. They're pattern recognition receptors, so they notice patterns that are similar on microbes, and they are ultimately important for recognizing the several different patterns that are always the same on a microbe. So, for example, TLR4 recognizes LPS, and LPS will always activate TLR4. TLR2 is activated by a few things, but the one I want you to know is peptidoglycan. And peptidoglycan is really thick. There's a thick layer of it on gram-positive bacteria. So you see an activation of TLR2 through gram-positive bacteria and an activation through TLR4 through gram-negative bacterial infection. TLR3 is important in response to viruses. And so here showing you some of the innate immune receptors, and so there's many other ones than those three that I've talked about. There's even some that respond to yeast that can have effects on changing types of cytokines that are released, which is kind of neat. But the ones that I told you about were the TLR4, so it can recognize and respond to LPS, and TLR2, which can respond to peptidoglycan on gram-positive bacteria. And so any of these signaling, what they do, the end goal, I forgot to mention, on the endosome is where you find the ones for, here's another endosome, for TLR3. So you have the activation of the TLR3 in the endosome, and that's key because you would find virus inside the cell, not so much outside the cell. So these activate signaling pathways which will change the transcription of genes in the cell to release cytokines. And so depending on the type of pathogen, you may get a different skew in the different types of cytokines that are released from that cell. And that can shape the immune response to be a bit different depending on the cytokines that are made. For example, you'll get a lot of IL-1 beta, you'll get TNF and IL-6 from bacterial infections, and from viral infections you'll get a lot of type 1 interferons. So when are interferons released? They are released after virus infection. And so viruses will bind to host cells, and some of them will be able to recognize that they are virally infected, and they'll release interferons. And interferons are cytokines that are released from virus-infected cells, and they can act on nearby cells that have interferon receptors to warn them and say, I have been infected, protect yourself, and induce this antiviral state. So what they do is they make these proteins, these antiviral proteins, so if they ever do become infected with a virus, the virus won't be able to replicate due to these antiviral proteins. Where do T cells and B cells mature? So both of these cells, these are lymphocytes, they are generated in the bone marrow. The B cells mature in the bone marrow, but the T cells mature in the thymus. So T cells mature in the thymus and B cells mature in the bone marrow. The bone marrow would be found in the middle of the bone, and the thymus is up here above the heart. So there are primary organs and tissues and secondary organs and tissues in the immune system. The primary organs and tissues are where the cells are generated, so where hematopoiesis is occurring. And the secondary organs and tissues are where antigen presentation occurs, so where the lymphocyte comes in contact with antigen. And examples of those would be the spleen and lymph nodes. How does the innate immune system activate the adaptive immune response? So an important or key cell here is an antigen-presenting cell, and a professional antigen-presenting cell is the dendritic cell. It's important in finding things in the periphery and presenting what they found to the adaptive immune response. So here on the left, it's showing you an antigen-presenting cell presenting to a T helper cell. So this presentation is most likely to occur in the lymph node or in the spleen, depending on if it's a blood-borne pathogen, it would occur in the spleen. If it's a pathogen that occurs in the periphery, so in the skin or different parts of the body, it'll go to a local draining lymph node to present what it's found. So T cells don't bind to just antigen on their own. They have to recognize antigen in the context of MHC. And T helper cells bind to MHC class 2, whereas cytotoxic T cells recognize antigen in the context of MHC class 1. So in order for a T helper cell to get activated, it needs to come in contact with antigen in the MHC class 2, and it also needs CD4, which recognizes this MHC class 2, and it needs a co-stimulatory signal. So the co-stimulatory signal from the antigen presenting cell, for example, it would be B7 molecules, B7, I can't write with this thing very well, and they interact with a molecule on the surface of the T helper cell called CD28. So you should know that this interaction is required, and this would be step 1 or signal 1, and this co-stimulatory molecule interaction is signal 2. So this is signal 1 and this is signal 2, required for this T helper cell to be activated. And when this T helper cell gets activated, it could become many different types of T helper cells, but I'm not going to talk too much about that because we didn't have time in class. But I do want you to know that the T helper cells, when they get activated, they need signal 1 and signal 2, and they get activated to proliferate. So there'll be more of them. And they also generate these memory cells, so the memory T cells, so they can be around for another time. Once they're more of the same one, so a T cell has a T cell receptor that only binds one antigen, and when that particular one gets activated, it's going to proliferate, so there's more of them. So you know you only want to make more of this T cell, this T helper cell, when you have that particular microbe around, and it's going to help the B cells get activated. So that's one of the things that cytokines released from the T helper cell can do, can have an effect to activate B cells. And so how that actually happens, we'll talk a bit more about B cell activation, but the B cell needs to be activated by antigen. So this would need to be the B cell receptor, which is the membrane-bound immunoglobulin, would need to bind antigen, and it would also need to then take it into the cell and break it up into little pieces and present on the surface of that cell in MHC class 2 to the T helper cell. And so that's part of the specific interaction that occurs. It'll present antigen. So this is signal 1, the antigen binding to the B cell receptor for this B cell activation. And signal 2 comes from these cytokines that are released from the T helper cell. And so cytokines they have a very short half-life, so they need to be close in order for this cytokines to have an effect on this B cell. And so they're close when the T helper cell binds to the B cell with its T cell receptor, so the TCR, and it interacts with MHC class 2 and antigen. So my T cell receptor. So that's one of the things that T helper cells can help by doing, is by activating B cells to ultimately, the goal is to secrete antibody. The other thing that they can do is make cytokines that are needed to activate the cytotoxic T cell. So cytotoxic T cells need 2 signals. They need one signal from recognizing antigen, and they recognize through a T cell receptor with CD8. CD8 recognizes MHC class 1, and they recognize a specific antigen. And then they also need these cytokines to activate cytotoxic T cells to be able to then, so they helps them proliferate, so there's more of them, and they can help them be good effector cells, meaning that they can actually go out and find infected cells. So they can find infected cells, and how they find them is they recognize the same antigen that they were presented over here that's being presented on MHC class 1 on that infected cell. So the T cell receptor on the cytotoxic T cell has to recognize antigen in the context of MHC class 1, and that's when, if it does that, it'll release these perforin and granzymes, which poke holes and kill this host cell, which is infected. So T helper cells are needed to make cytokines to activate B cells, and they are also needed to make cytokines to activate cytotoxic T cells to function properly. So this is looking closer at the activation of B cells by T cells. So this is an antigen-presenting cell, which presents to a T helper cell, which is naive. So it needs to be activated in order to function. So this is looking a little bit closer at some of that activation. So the T cell receptor on the T helper cell recognizes antigen in the context of MHC class 2. And what helps recognize that MHC class 2 is CD4 on that T helper cell. So that's signal 1 right there. So that's signal 1 to activate that T helper cell. Signal 2 comes when the co-stimulatory molecules bind. So the B7 molecules, which are CD80 or CD86 on the antigen-presenting cell, interact with CD28 on the T helper cell. And that causes the cell to proliferate. So it expands that type of T cell. So you have more of them. So you only want more of them when they're needed, and so that's why you activate them to have more of them. And so once you have more of them, they release the cytokine IL-2, which helps, which is needed for the proliferation and expansion of these cells. So this T helper cell, which has been activated, can go out and interact with B cells. And so the B cell needs 2 signals to be activated. The first one comes through interaction of antigen with its membrane-bound immunoglobulin. So this little yellow thing is the antigen binding to the immunoglobulin. That's signal 1. That actually comes into the cell, and it can be the antigen can be chopped up into little pieces and then can be presented on MHC class 2, which is here, to a T helper cell. And so this T helper cell is recognizing antigen in MHC class 2 on the B cell. B cells are antigen presenting cells. I told you that before and a few slides ago. And what recognizes that is this CD4 molecule, recognizes the MHC class 2. So when this happens, it will release cytokines. So there's several different cytokines that the T helper cell can release, which will help that B cell expand, so proliferate, so there's more of them. And you also get memory B cells. The plasma cells are the ones that make and release a lot of antibody. And so the antibody that are released here would be the same, specific for the same antigen that was on the surface of that B cell. And they would be able to bind to that antigen and try to clear or get rid of it. What are the functions of antibodies? So there's a few different ones. Precipitation and agglutination, they basically clump the microbes or the antigens together so that they're easier to clear. It's easier to find one clump of microbes than to go out and search for each individual microbes, so that's helpful. Particularly IgM and IgA are good at agglutinating, but IgG can also do it. Opsonization is only able to be done by IgG. IgM and IgA can't opsonize because they don't have their Fc regions available. Opsonization is a term used for enhancing phagocytosis. So antibody is an opsonin because it enhances phagocytosis. And there's antibodies bound to this microbe here, and they're binding to, on the surface of these phagocytic cells, it's not drawn here, but there are Fc receptors. So they're binding to receptors on the surface of the phagocytic cell, recognizing that the antibody is bound to an antigen, enhances the phagocytosis of it into that phagocytic cell. You can also have complement fixation. So we talked briefly about complement, and one of the things I want you to know is some of the outcomes of complement, and one of the outcomes are that it can activate and lyse bacteria. We also talked about how it can influence inflammation. So there's anaphylatoxins that are released that can influence inflammation and recruit other cells to the area. And what's another thing? Poke holes, inflammation. I forget. I'll see if I remember. And enhance opsonization. So there's complement proteins that can also do that. So C3b is the one that we looked at in class. That is also able to act as an opsonin. Antibodies are also able to act to neutralize or block microbes from attaching to cells and binding to them and getting in and causing infection. So they can neutralize virus. What's different the second time you come in contact with the same microbe? So the difference with respect to the adaptive immune response is timing and how quick it can respond. So your book says about 10 days it takes to get antibody the first time you come in contact with a pathogen. So you get IgM around 12, really high levels, 12 or so days, 12 and a half days after coming in contact with the microbe. And then it takes even more time to get IgG. So IgM is really good at agglutinating and activating complement. IgG can enhance phagocytosis and act as an agglutinator and neutralizing microbes as well. So it takes about more than 2 weeks to get a good amount of IgG produced. However, the second time you come in contact with the same microbe, you get a lot of IgM and IgG shortly after 5 days of a second exposure. And these levels are higher because you have memory cells that were generated the first time you came in contact with that pathogen. And so you'll have those memory cells there ready to divide quicker once activated again. So this cuts down the time that you would be sick, or you may not even know you're sick at all because it could lead to a subclinical infection whereby you don't have a high level of the microbe multiplying and dividing in your cells or in your body, and so you don't even know that you may not have symptoms of that infection. How does HIV interfere with the immune response? So we looked at an example in class, and particularly HIV infects CD4 positive T cells, so that's the main cell that it will infect. And reducing the number, so it's grayed out even more here, then reducing the number of the T helper cells make it so that you have less possibility to generate cytokines which are needed to activate cytotoxic T cells and are also needed to activate those B cells like we talked about. So if you don't have these T helper cells, so you have a reduced number, you would have less chances of activating the cytotoxic T cells, and they're needed to kill virus-infected cell. So they kill virus-infected cells. It's going to take forever. Kill virus-infected cells, and they would also, these cytokines would be needed to activate this B cell to produce a lot of antibody. So this one doesn't kill virus, but it kills virus-infected, sorry, I want to finish it, infected cells. So make sure you are familiar with the idea that cytotoxic T cells don't go and kill pathogen directly, they kill host cells that are infected with pathogen. So if you don't have these cells, you don't have the activation of the cytotoxic T cells or antibody. So you effectively shut down the adaptive immune response, kind of at the heart of it. I hope this helps. I'm going to, it's going to take me at least 2 or 3 more days to get the other video I spoke about on, because I need a stylist to get it to work. But I hope this helps to get you started studying. Let me know if you have questions via email.